Rosalind.
Good evening, everyone. We will begin the session with a prayer by Dr. Rita Jaira, Professor, Department of Zoology.
I kindly request each one gathered to assume a prayerful posture. To you, O Lord, I offer my prayer. In you, my God, I trust. Teach me your ways, O Lord. Make them known to me. Teach me to live according to your truth. For you are my God who saves me. I always trust in you. As we have gathered together for the online lecture, I especially pray for our speaker, Dr. Soumen Basak. Shower your abundant graces on him, enlighten him to make his talk a resourceful and meaningful one. Come, O Holy Spirit, empower our principal, Dr. Sister Rosie Joseph, the secretary of our college, Sister Susan, and the management. I pray for our collaborators, Dr. Meenakshi Munshi, the coordinator, Ms. Jyoti, for all my colleagues, student participants, and specially pray for our student volunteers of this program. May the entire academic exercise bear fruit in good time. Mother Mary, shower your maternal protection on each one of us and continue to guide us. All this we ask in the name of our Savior, Lord Jesus Christ. Amen. Thank you.
Thank you for the beautiful prayer, ma'am. I now invite Dr. Satyabama, Assistant Professor, Department of Botany, to welcome and introduce the speaker of today's session.
A very good afternoon to one and all present here. With immense pleasure, as we are celebrating the semi-sesquicentennial year of our independent India and Stella College together, we are proud to present a lecture on regulating inflammatory immune response, learning from Math plus Bio through Science Setu program. It's my privilege to welcome our beloved principal, Reverend Dr. Sister Rosie Joseph. I wish to place on record her total support in organizing this lecture. I place on record my sincere appreciation to our college secretary, Reverend Sister Susan, and the vice principals for all their guidance, wishes, and prayers. With great pleasure, I welcome the resource person of the day, Dr. Soumen Basak, Staff Scientist, System Immunology Laboratory, National Institute of Immunology, New Delhi, who accepted to share his expertise to our students. You are most welcome, sir. We are keen to hear from you.
Thank you very much.
A special note of welcome to Dr. Jyoti Singh, PRO, National Institute of Immunology, New Delhi, who is a pillar of strength towards this program. I also welcome my colleague, Dr. Rita Jairaj, Associate Professor, Department of Zoology, and also my dear students for their active participation. I wish you all a fruitful and enriching experience and have a pleasant day. I now introduce our speaker of the day, Dr. Soumen Basak. Dr. Soumen Basak is the staff scientist grade 6 in the System Immunology Laboratory, National Institute of Immunology, New Delhi, India. He has contributed towards spearheading system modeling studies in India. Dr. Soumen has specialized experience and discovered a novel signaling crosstalk circuit which integrates signals from inflammatory substances and immune differentiating cues for tuning physiological immune responses and whose all functions promote cancer cell survival. His research will inspire novel therapeutic intervention strategies that target signal cross regulatory mechanisms in human ailments. He was a postgraduate researcher in the Department of Chemistry and Biochemistry in the University of California. Then he entered National Institute of Immunology, New Delhi as staff scientist grade 4 in the year 2010. Slowly, he got promoted and reached the grade level 6 as staff scientist in the same National Institute of Immunology, New Delhi. That's great, sir. Dr. Soumen is the recipient of several awards and obtained various prestigious positions. To mention a few, in the year 2010, he received the Intermediate Fellowship from the Wellcome Trust DBT India Alliance. In 2017, he was elected as a member of Guha Research Conference. In the year 2018, he is the recipient of the Faculty Award from the AGN Conference held in Greece on Pathway Network and Systems Medicine. In the same year, he also received the National Bioscience Award for Career Development from DBT India in recognition of the research in the area of immunology and cell signaling. In the year 2019, he received Shanti Swarup Bhatnagar Award from CSIR India for research excellence in biology. This year, that is 2021, he is elected as a Fellow of the National Academy of Science India and Indian Academy of Sciences. To add a feather on his cap, he has received 3 major extramural research funding. He has many scientific publications too. To mention that he has he has published in 5 reputed journals. That is really inspiring, sir. And girls, his CV is too long. Due to time constraint, I think I can stop here. He is such an eminent person. I call him now to share his expertise. Over to you, sir.
Dr. Basak, kindly unmute and speak. We are not
Yes,
able
I
to
noticed
hear.
that.
Thank you.
Thank you, Dr. Rita, and thank you, Dr. Satya. This is
Thank
really
you.
pleasure to be here today. And I also have to thank Jyoti to put this together on behalf of NII. And it's really a wonderful thing. Like, we are celebrating 75 years of our independence. And here I am giving talk in a college which I believe came into being on the date of India's independence, right? 15th August 1947. Am I correct?
Yes, sir. Yes, you're correct.
This is really humbling, and it really I noted this interesting fact about your otherwise very well-known college. Let's see if I can make this next 1 hour enjoyable for us. I mean, we will go into research area which might appear a bit complicated, but our whole ambition would be to have a take-home message from today's talk and sort of take it forward in future so that it can help us to continue to have aspiration to learn more. So let's see where we are. I mean, this is National Institute of Immunology. Here you see this nice little building. Just a minute, yeah. So this is this nice little building, National Institute of Immunology, and my laboratory is unfortunately still in the basement. And this is my research group. The picture is a bit older. We couldn't take a group picture for quite some time because of COVID-related restriction. That's a pity. But we have around 10 to 12 students, and our group is mostly consisting of PhD students. But we do adore when master students from all across India come and do work in our laboratory as a project trainee for 6 months or so. But that's us. And what do we do? I mean, we're going to really talk about this regulating inflammatory immune response, learning from math plus bio. It looks very complicated. Let's see if we can simplify and can have an enjoyable experience. First question is, why do we study immunology, right? We're talking about inflammatory immune response. Why is it important to know about immunology? My life has become much more easier after COVID. It used to be a bit complicated. I used to have a lot of things I have to tell them. And this slide was prepared before COVID. Of course, after COVID, you need to learn immunology so that we can potentially handle threats for mankind. But let's see what is it. You know how many viruses are there on Earth? It's 10 to the power 31. So if you put them next to each other, we're talking about 100 million light years. And one third is land, the rest of the world is Earth is ocean, right? And then the number of bacteria in the ocean is 10 to the power 29. It's mind-blowing. It's 100 million times more than the number of stars we see in the known universe. And you take a spoonful of soil, how many bacteria do you have, you think, in this? 10 to the power 9. Same as the number of human living in continent Africa. And out of these microorganisms, I agree, not all of them are harmful, but we are really deep inside an ocean full of microorganisms. And out of these, at least for today, only around 1,500 of them are human pathogen. They cause disease in human. They cause painful disease in human. So we are all around us, a lot of microbial entities. And then when you look inside, as you're born and you're growing old, by the time you're old, you will have 100 new mutations in your genome, in some tissue or the other. 100 new mutations, many of them, trust me, will be in tumor suppressors gene, meaning those mutations will cause tumor. So, indeed, a study was done using volunteers, and it was found that we have many precancerous lesions when the study was done in, mostly with breast tissues. Many precancerous lesions than the frequency at which you get breast cancer. Similarly, it appears from inside, we are always exposed to some threats and someone is saving us. Who is it? That's why you should learn immunology. That immune system protects you from these microbes, despite you're deep inside this ocean. You're not sick every day, and that you have those tumor threat, mutation threat of developing cancer, and you're not getting cancer with that high frequency nonetheless, is because of immune system is protecting you. And when it comes to immune system, probably I looked into it. Some of you are probably second year undergrads, the third year undergrads, those who know a little bit of immune system. There are two arms really, innate immune system and adaptive immune system, right? And innate immune system is, as it says, innate. It came early during evolution. Even in arthropods, you'd see them, nematodes, you'd see them, molluscs, you'd see them. Adaptive immune system, the immune system that you talk about when you say B cell, T cell, these are all adaptive. They came much later. They came when early vertebrates evolved on Earth. So there was something protecting us even before, or protecting life, even before B and T cell arrived. And that's innate immunity. And this is a drawing. It's not really a formal data. You see what you have in x-axis is time. Minutes, hours, days, weeks. Do you see them? Right? And in y-axis, we are looking at the activity of various things. You see, you have a barrier, you have a physical barrier, skin, that protects you, right? Otherwise, your body will be exposed to microbes. And let's say there is a breach in the physical barrier, microbial entity like a bacteria has entered your, let's say, little cut in your hand. And that microbial entity immediately triggered, the first thing is an inflammatory innate immune response. That's an innate immune response. And that then triggers that happens within hours and continues for a few days. That's the first response you give. I got bacteria, I give innate immunity. I get inflammatory immunity. That is the same thing. We'll come to that in a minute. And within a days, this innate immune response then triggers adaptive immunity. That is the B cell, T cells. That continues for weeks and months, so as to make sure not a single bit of bacteria or pathogen is left in your body. But initial response is innate immunity. And this is again a drawing. A drawing shows the yellow line, how number of microorganisms, if they enter the body, their number is regulated over time. You see this yellow line? It goes up initially, and then it slowly falls down in a bell-shaped curve almost. Is because of our innate immunity and all this, and adaptive immunity. And then if you lack adaptive immunity, you see green curve, that the fall down is not happening. But if you lack innate immunity, see red. It's all over. You can't control anything. From the very beginning, then there will be a rapid increase in the microorganism number, and that would lead to fatal disease. So that's why I say that this innate immunity is not only came early during evolution, but it's also dominant. It's a dominant role it plays in checking infections. So what is innate immunity? I'm going to talk about few components. Some of these you will learn maybe in a year or maybe a bit more than a year. It's a, we call it anatomic barrier, right? Skin. It's an anatomic barrier. It protects us from microorganisms, no question about it. But there are these chemical, physiochemical barriers, like stomach pH. When you ingest, when you bite a fruit or raw vegetables, they do have microorganisms in them. And when you bite, they go into your stomach, but the stomach pH is very low, acidic. It kills those microorganisms while degrading the food particles into smaller pieces so that we can ingest. So these are innate immunity. They are with us. It doesn't matter what microorganism we are attacked with. Is it virus? Is it bacteria? What kind of bacteria? It doesn't matter really. Similarly, there is another innate immunity component called inflammatory immunity. And this is where macrophages or tissue resident cells, they play a role in triggering this inflammatory immune response, which is initially going to protect you from those microorganisms and limit the microorganism growth in the infected tissue. Inflammatory immune response, just like any innate immunity, it is not specific for a pathogen. It will be triggered by anything and everything. Let's see what we have. Let's little bit learn about inflammatory immune response, and then we'll get into the mathematics. That is our job today. So this is what I'm showing you. This is blood, and these cells, these are endothelium, blood endothelium, right? Blood vessel. And this is the tissue beneath it. And a pathogen, a bunch of pathogen has entered, it started replicating there, pathogen numbers are going up. What will happen? The tissue resident cells, your skin cells, your endothelial cells, your epithelial cells, and then there is this specialized, very important cell. These are macrophages. They will sort of sense that there is a pathogen, there is a foreign particle. They will sense, they understand that there are pathogen-derived substances. They recognize them. They have protein that they express on the surface. We call them receptor proteins, right? These receptor proteins will recognize that there are pathogens, there are pathogenic substances, they are not belonging to my body, and that these pathogen receptors are called toll-like receptors, TLRs. And it was originally discovered in 1993 in Drosophila. They used to call them in Drosophila melanogaster. These were discovered, and they began to call them toll receptor. Later, they found similar cell surface protein, otherwise called receptor, do exist in human cells as well. Then they began to call them toll-like receptor. So in Drosophila, you have toll receptor. In Drosophila and mammalian, you then began to call them toll-like receptor. And these three fellows, you see, they got Nobel Prize for discovering this toll receptor. Bruce Bartlett, Ralph Steinman, and Jules Hoffman. They together discovered these cell surface TLRs that recognize when pathogen is there in the tissue. So, okay, pathogen has entered the tissue, and these are recognized by these toll-like receptors. Now, these toll-like receptors will send signal into the nucleus so that immune response genes are expressed. So, you have receptor on the cell surface, and the nucleus, if I can understand that you guys know, the nucleus you have DNA, and that DNA should be transcribed for allowing cell to respond to external environment, right? So you have cell receptor and you have nuclear DNA, and there is this signaling pathway. That is a pathway we will discuss a bit. There is some protein inside cell that will relay this signal so as that a particular transcription factor that is important for inflammatory response is activated in the nucleus. That allows inflammatory or immune response genes that are there in the genome to be decoded for the mRNA to be synthesized. Now, basic some transcription slide I put for you guys to understand. So, transcription of a gene leads to mRNA, that leads to protein. These are very important for cell to respond, and that totally characterize what immune activity we have. Now, what you have in a gene? You of course have a code ORF, right? Open reading frame, which will, if it is transcribed into mRNA and translated, this will lead to protein. But the gene has this upstream promoter element, right? Where RNA polymerase 2 should bind, but it cannot bind on its own. It needs this general transcription factor, let's say TBP, data binding protein. It's a general transcription factor. It binds to its cognate DNA sites, TATA, we call it Tata box. It's a DNA sequence that is rich with T and A, where TBP sits. And when TBP sits, it recruits RNA polymerase, and that may allow gene to be transcribed. But you know what? Immune response genes won't get transcribed just because you have TBP recruiting RNA polymerase 2. It requires immune related transcription factors to be there. One is NF-kappa B. This is an NF-kappa B is a transcription factor. It binds to similarly NF-kappa B site. This is a DNA site. It is a bit GC rich site. And there are always in immune response genes in the promoter. You will have a Tata box where TBP will bind recruiting RNA polymerase, and next to it, you will typically have an NF-kappa B DNA site where NF-kappa will bind. And now NF-kappa will kick the RNA polymerase, so RNA polymerase will start rolling, making mRNA. That leads to those mRNAs are important. Those mRNAs encode proteins that are important for immune response. So you need immune signaling. What you get there is that there are cell surface receptors. If they are active, if they are engaged, if they sense pathogen receptor, NF-kappa B will be activated. It will be in the nucleus and it will bind to NF-kappa B site and will help expression of immune response gene. Now, how this signal is induced factors? NF-kappa B was not active. Once the TLR was active, NF-kappa B became active. How is it activated? How the signal induced factors are controlled? Why are they signal? You have basic or general transcription factor that are there always in the nucleus. And then I'm saying there are signal induced factor. Once there is pathogen, TLR sends them, that cell surface receptor sends them, this transcription factor that I'm calling signal induced factor will be activated in the nucleus. How? There are three scenarios, right? One, this transcription factor is not present in the cell. Once there is a signal, these transcription factors are produced by per se, and then they act on their cognate genes, target genes. Scenario 2, transcription factor is present even in the nucleus, but it needs some post-translational modification. You guys must have heard of protein phosphorylation. Some transcription factor needs to be phosphorylated so as them to bind to DNA and activate genes. In the absence of those phosphorylation event, they will not be active, and pathogen recognition may lead to their phosphorylation leading to their activity. And the third scenario is today's scenario. There are transcription factors that are already present in the cytoplasm, and when pathogens are recognized by cell surface receptor, they trigger nuclear activity. They promote nuclear translocation of this particular transcription factor like NF-kappa B. NF-kappa B is always in the cytoplasm. Once pathogen is recognizing, being recognized by cell surface receptor, NF-kappa B, I will tell in a minute how, NF-kappa B rapidly translocates into the nucleus where it binds to its cognate gene, the NF-kappa B site, and thereby triggering, thereby provoking adjacent RNA polymerase to initiate transcription. And this is how immune response genes are transcribed. So let's see what is going on here. Again, coming back to the tissue, remember bacteria and being recognized by TLR. Now we know there is this NF-kappa B transcription factor. There is a particular pathway which will allow NF-kappa B to go into the nucleus. Once it is in the nucleus, it transcribes genes that are immune response genes. Who are they? For example, antimicrobial protein. There are antimicrobial proteins that are even expressed by Drosophila. They, bind to bacterial membrane and lyse them. There are phagocytosis. I will show you a very nice video today. Hopefully it will work. But these macrophages will engulf bacteria and then take them in their lysosome. Lysosome, if you have heard, lysosome is a acidic compartment within cell where the bacteria will be degraded, thereby inactivated, right? And then third, don't undermine it. This tissue resident cell will make a lot of proteins termed chemokines and cytokines. These are messenger. Macrophages or tissue resident cells are good in bacterial killing, not very good. This messenger will drag these neutrophils. They are deadly assassin. They have granular proteins. You see, they have full of granules. Those granules will be released. These granules are full of deadly protein. They are antibacterial peptide. They are proteases. They are myeloperoxidase that causes reactive oxidation of bacteria. Full of deadly protein. So the first work is, the local cell will release antibacterial peptide or cause phagocytosis to clean up bacteria, but they will also release chemokines and cytokines which will drag neutrophils, which will further augment the process of bacterial clearance. If you see, this is the thing. I'm going to show you this. Let's see if it works. Yeah. We are gonna
Neutrophils are white blood cells that hunt and kill bacteria.
So this is neutrophil.
In this spread, a neutrophil is seen in the midst of red blood cells.
These are red blood cells.
Staphylococcus aureus bacteria
And
have
this
been
is a
added.
small tiny bacteria.
The small clump of bacteria release a chemoattractant that is sensed by the neutrophil.
Now neutrophil is
The
chasing them.
neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
Now they have engulfed the bacteria and destroyed it, putting it in the phagocytosis, through phagocytosis, putting in the lysosome. Now, question is, neutrophils and macrophages, they express a lot of, oxidative stress causing reagents or chemokine, cytokine. They are deadly cells. And this is innate immunity. They are not going to cause harm to this bacteria, but also harmful for the host. The tissue cells will also die because of reactive oxygen species. Tissue cells will also be harmed because of neutrophil activity or macrophage activity, chemokine, cytokine. So the whole point now I'm going to come to this second aspects of inflammation, that it must be contained after some time. Inflammation is caused by NF-kappa B. We said the NF-kappa B goes into the nucleus, otherwise it is in the cytoplasm, right? And that leads to all this inflammatory gene expression or immune response gene expression. Now we are saying that immune response gene must be controlled after certain activity. So there must be induction of their activity, and there should be, after activity is reaching a peak, it must rapidly fall down. So who causes this activity? NF-kappa B. So in other words, one can say NF-kappa B activity should be kinetically controlled. And now I'm going to really talk about this a little bit. It will be complicated. This is NF-kappa B. This black and white is NF-kappa B. It is typically in the cytoplasm. Okay. When the TLR is activated, let's say LPS is lipopolysaccharide. It is typically present in bacteria. If you treat cells with TLR, and it actually acts through TLRs to activate cells. So once you treat cells with TLR LPS, what will happen? This NF-kappa B is typically in resting cell bound to inhibitor protein called I-kappa B. And that keeps the NF-kappa B protein in the cytoplasm. Once you treat cells with bacterial substance, this I-kappa B gets degraded, leading to NF-kappa B release into the nucleus, where it activates dozens of immune response genes, those phagocytosis genes, those antibacterial proteins, chemokines, cytokines. In addition, NF-kappa B also activates its own inhibitor, I-kappa B alpha. So what is this? In resting cell, NF-kappa B is bound to I-kappa B. Once you trigger them to pathogen molecules, then I-kappa B is degraded, NF-kappa B goes into the nucleus, and the bottom one is showing you the activity curve. Y-axis is NF-kappa B activity and x-axis is time. So NF-kappa B, once you treat cells with pathogenic molecule, NF-kappa B is activated, goes into the nucleus. So I'm basically plotting nuclear NF-kappa B. So you get a peak. Once it is in the nucleus, NF-kappa B then activate dozens of genes, but it also interestingly activates its own inhibitor, that I-kappa B. So what will happen? Once the inhibitor is activated, it will now try to inhibit NF-kappa B, right? There will be a post-induction termination of NF-kappa B. So there is activity, and then it rapidly falls down because of I-kappa B alpha mediated negative feedback. So we got it. We said inflammatory signaling must be contained after some time, and there is appearing to be a negative feedback that is very good at containing activity post-induction. So there is a negative feedback within the NF-kappa B system. Now question is how you measure NF-kappa B activity? An experimental approach. NF-kappa B, when it is nuclear, we can measure it by this. I mean, I'm going to really detail it because you guys read these experimental protocols and these are within your curriculum. So I'm going to show you something very interesting. So you treat cells in a time course. You treat cells with LPS, which is a TLR ligand, right? And then collect cells and then make nuclear extract. And what you do in the nuclear extract, you add a small oligo, a DNA, double-stranded piece of DNA, small DNA, which has NF-kappa B site, and of course it's radio level. Okay, you incubate them. All right, and then load it in a native gel. You see, if NF-kappa B factors are there in the nuclear extract, they will bind to this double-stranded DNA because the double-stranded DNA has NF-kappa B binding site. If in your nuclear extract there is no NF-kappa B dimer, then double-stranded DNA will not be bound by any protein. So when you load the gel, if there is no NF-kappa B in your nuclear extract, the double-stranded DNA will migrate freely. if there is some NF-kappa B protein in your nuclear extract, then it will form a complex with double-stranded DNA, slowing down its migration through gel. And you put an autogram, you will get an autoradiogram where you will see that NF-kappa B binding activity will leading to a shift in the DNA migration. So it will typically migrate here, but if NF-kappa B is present, you will see a shift. You will see a shifted complex. This is what we call EMSA, electrophoretic mobility shift assay. And I'm showing you the real life data. I'm only showing you the shifted complex. I'm not showing you the bottom of the gel where the free probe runs. You see, you treated cell with LPS in a time course 0.51, 3, 5, 8, 16, and 24 hours. After LPS addition, you are collecting cells and making nuclear extract individually. And then you are checking out this nuclear NF-kappa B activity. It goes up very rapidly. Within 1 hour, you see a very strong nuclear NF-kappa B activity in the nucleus. Typically, in this resting cell, you do not see NF-kappa B in the nucleus. Once you treat them with LPS, you see NF-kappa B accumulation in the nucleus, and then you see slowly, but very, then you see very gradually this NF-kappa B activity fell down, and that is because of negative feedback. Now, what is the problem with the negative feedback? You have PAMs, you have pathogen-associated molecular patterns, and you get NF-kappa B activity. But remember, this is very important to remember that there is a problem with that negative feedback, tight negative feedback. Because you have this NF-kappa B activity is a dynamically controlled activity, right? You have going up and falling down. But if it, and this activity is very important for expression of chemokines, cytokine, antibacterial peptides. If the activity, because of strong negative feedback, it can lead to insufficient NF-kappa B activation. It comes up and very rapidly fall down without showing much of a gene activity, without showing much of a gene expression that NF-kappa B is supposed to do once it is in the nucleus. Because of negative feedback, it fell down too early. And that would lead to immune deficiency. So the question is how to sort of control this in physiology, where you have a negative feedback, but you need a real accurate NF-kappa B persistence in the nucleus so that you have all the inflammatory genes expressed, immune response genes expressed, and you can protect yourself. And this is the third part of the talk, and there will be a fourth part and will be done. So I said you that, bacteria, you have pathway 1, you get a response, and you have some other thing, let's say neuronal signal, you have pathway B. So there are cellular pathway. We talked about NF-kappa B pathway just right now. With that pathway that we saw, we talked about I-kappa B and LPS. This pathway we talked about, right? This pathway is called, I'm going to take you back a little bit. This pathway is called canonical NF-kappa B pathway. And LPS or any other TLR ligands degrades I-kappa B, releases NF-kappa B activity into the nucleus. And this is subjected to negative feedback control. That is called canonical pathway. Now you can say there is another pathway which controls some other aspects of biology. So you have different pathway and controlling different transcription factor. Life was easy. But this is an interesting slide. I will take a minute for you to understand. This is very interesting slide, which will lead you to understand why we are here. So there is this gene A. It makes protein A, and protein A has an activity. Let's say it's a kinase. So, gene A encodes protein A, and that is a kinase. I am measuring kinase activity. And, I hope you guys will read sooner than later. There are this technology called siRNA technology where you can make gene A non-functional. Okay. So what you have in this graph is this x-axis. You have 24,000 points. Each of them represents one or the other Drosophila ORF. So one or the other Drosophila ORF was inactivated. And in y-axis, you are measuring, let's say, activity of the kinase. Activity of protein kinase, this kinase A will be determined by, activity of kinase A will be determined by, gene A. So when we look into this graph, of course, there will be one hit where when I inactivated gene A, kinase activity fell, like the extreme left side of the graph. And there could be some inhibitor of the kinase. I understand extreme right side of the graph. What I do not understand that all genes shows a non-zero value, meaning it appears that all of the genes that Drosophila has some contribution, maybe low or high, some contribution in determining kinase activity of the protein A. And this is the concept of systems biology, that you are no longer talking about that one pathway or one protein is controlled by one pathway, one kinase, right? It appears these pathways are all interlinked and not individual pathway that determines cellular response, but a combination of pathways interacting with each other, a system consisting of interlinked pathways determined system. And this is the arrival of system biology, where we are saying no, not only one receptor, one canonical pathway, and one transcription factor is going to call the shot. Actually, these pathways, there are many pathways in the cell, they are all linked, and that together gives cell system that response. And that is where the dynamics is controlled. That is what the temporal profile or kinetic profile is controlled. And what is it? And then that is what the arrival of system biology. This is system biology. Next 2 to 3 slides, we'll remember this forever. So these are 4 objects I am showing you in the slides, right? Glass, inert gas, tungsten, and metal cup. And it look like very different 4 different things together in one slide. Then I show you that you can put them together in a particular manner that we call topology in some formal language, so that you get an electric bulb. It still appears that way. I understand it's intuitive. Then you put input energy, you get light. That is emergent. I would never have thought of light when I had seen glass, tungsten, argon, and metal cup. So it appears that when you put together components, then the system acquire properties that is more than the sum of the properties of the component. You can't explain light by looking at glass or tungsten or inert gas or metal cup. Together, putting them together with an interesting input, that is energy, you get something that is not explainable by each component. So that's called emergent properties, and we're basically saying that this is what it is. That this is a dynamical control. You need to go up and then fall down, but not rapidly, not too late. It has to be very accurately controlled so that you do not get disease, but you can clear pathogen. That control, that timing control, we are basically saying is because this is an integrated system where many pathways are interconnected and they together fine-tune this property. And why is it even important in inflammation? This is where we were. A tissue resident cell, we had pathogen. These red dots are pathogen, and this is a tissue resident cell. Now, if you think of it, in this tissue, and these microbial pathogens actually activate the canonical pathway, and which is under negative feedback control. And in these tissues, you also have many other cell types that actually secretes cytokines that activate another pathway called non-canonical pathway. So now we're talking about 2 pathways. Canonical pathway, which is under negative feedback control, and his duration of the canonical NF-kappa B activity must be controlled very tightly, very accurate, in a context-specific manner, tissue-specific manner. This has to be achieved. And there is this in the tissue, there is another thing that is the non-canonical NF-kappa B pathway. What we have learned that it has been reported these pathways are also linked. So this provides for a small system, right? There are 2 pathways, they are linked. Do you think that then this non-canonical pathway actually modulate this NF-kappa B response that are generated, this late activity, going little early, going little late, going little up, going little down? Is it because this non-canonical, canonical link together, is it happening? And how do then, actually now we are talking about 2 pathways, right? And they are interlinked. This will lead to emergent properties. Remember the bulb and the light? And these are non-intuitive. You would never think of light when you look at tungsten and argon. Similarly, canonical and non-canonical pathway together will give you properties, if it is linked, that are not so easily explainable just by looking at canonical pathway, whatever protein and cell culture, cell experiment you have done. How do you go and probe the non-intuitive property? And this is the last section of the talk. This is the picture you all remember, right? Here's the picture. This is an Apollo 11 mission, 1969. Historic day for mankind. And this is none other than Edwin Aldrin.
The first man to step foot on the moon.
Sorry? Yes, yes, absolutely. You remember this picture. See, everyone remembers it. Do you remember this picture? Nobody would. This is not on moon. This is on NASA research facility. It's nothing but a lunar excursion module simulator. It's a simulation mission. Thousands of simulations were done, and the results were acquired, analyzed in a mathematical algorithm for perfecting moon landing. When you have a complex situation, too many things are happening, probably want to go back to mathematics, right? This is the last part, biology plus mathematics. You see, what I'm showing you, I'm sure some of you have chemistry in your undergraduate, some of you do not. It's sad. Mass action kinetics is a property that controls chemical and some biochemical reactions. You have an input that I'm going to explain it to you within a minute time. You have an input that accumulates X. X is degraded with a rate constant alpha. So what will happen? This equation says changes in the X. X dot means dx dt, meaning rate of change of X. You have input that will accumulate X, right? And X will be degraded with a rate, with a certain rate, right? The rate constant alpha. So initially there is very little X, so X will rapidly accumulate. Eventually what will happen? Because a lot of X has accumulated, degradation rate of X will increase, right? And then eventually equilibrium will rate. So the amount of X is produced by the input and amount of X is degraded is equal. Now, let's make it a bit complex. X makes Y, Y is also degraded. Then you get another equation like that, right? Life is not difficult. How changing the Y concentration, you can actually write these equations. Don't worry about it. You don't have to go into the equations. See the theme. Now, let's say, negative feedback, right? NF kappa B is also making I kappa B, that gives negative feedback. They're going to give negative feedback. Let Y inhibit X. And you can write, of course, another set of equation, right? Because Y inhibited X, that rate constant is beta. Let's put it in some equation. I do not want to get into that. What I'm getting into is that there are a couple of rate constants now. Alpha, beta, gamma, delta. And then this is how X and Y will change over time. Remember, we said if activity is too early terminated, we have disease. If activity is continued for too long, we have disease. Activity duration must be very accurately controlled for us to give appropriate immune response. And now this is what it is. Alpha, beta, gamma, delta, and if you keep changing it, you will see that this is a very simple set of equation. Very simple system. All I'm trying to get home is this. I am going to change now alpha, beta, gamma, delta. See what is happening. A very simple system with a bit of differences in the combinations of the rate at which these reactions are occurring can leave it to any different possibilities of Y activity in the system. And this is what it is. When you say, okay, how long NF kappa will continue? Well, these are the all molecules. You do not have to go into this. Too many biochemical proteins are actually controlling the activity of the NF kappa B. We can write equations similarly. We can figure out the rate values, and we can actually, given too many equations, we can put them in a computer model and simulate how NF kappa B activity would be looking like and what could be the determinant of its changes, right? And that's what we do in this lab, in my laboratory, in the systems immunology laboratory, but system biology is done. This is a computational model we have. We will simulate, we will test those simulation results in cell culture. Sometimes we'll do animal experiments. Sometimes results appear to be true. Sometimes results are not matching up with what we see in the cell culture. We'll go back to the computational model and refine it. It's a never-ending loop. So make a stronger model which has predictive value. And this model says that. Now I'm going to really give you 2-3 minutes. This red line is how cell responds to TLR ligands. You have NF kappa B activity, falls down. And the model indeed said, if the non-canonical pathway is also activated, this can lead to changes in the late activity. Look at the black line. Model indeed said that NF kappa B, the non-canonical pathway, can crosstalk with the canonical NF kappa B pathway. And this is what we're calling NF kappa B pathway crosstalk. And that can change the late activity. It can calibrate. If you have a lot more non-canonical activity, this black line will go quite up. If you have little less non-canonical activity, the black line will go quite down. But that's how you can, in a context-specific manner, calibrate the late activity, calibrate the duration of NF kappa B response. And this is the experiment. Again, EMSA, same exact results we got. That LPS activity going up, falling down, LPS-induced activity. And when you give the non-canonical pathway, you see the late activity can be sustained to some extent. And that led to differences in the gene expression. These are the cytokines, IL-1 beta and RANTES. Remember I said chemokines and cytokines, that will bring neutrophils at the site of infection. Neutrophils are typically in the bone marrow. You have to give them signal, and that signal is IL-1 beta, RANTES. These are the chemokine, cytokine. That sends signal to attract neutrophil at the infected tissue where they can eliminate bacteria. And indeed, it appears that if you have non-canonical pathway, you have a little heightened expression of these chemokines, cytokines at the late phase post-treatment with cell treatment with LPS. And then modeling also said that these two pathways are linked because of a gene called NF-kappa B2, which had nothing to do with immune response. Initially, we thought. But this appears that indeed NF-kappa B2 knock gene is very important for immune response. What am I showing you now? These are mice infected with Citrobacter rodentia pathogen. And we are showing you that if NF-kappa B2 wild-type mice, if you infect them with pathogen, you see fine NF-kappa B activation in the infected tissue. It's exactly the way it predicted. That would lead to expression of inflammatory genes. Look at KC and MIP2 alpha on the right-hand side of the slide. But if you do not have NF-kappa B2 gene, which is nonetheless not a part of the canonical NF-kappa B pathway, you see NF-kappa B response is timid, gene expression is almost null, and that led to systemic bacterial dissemination. Wild-type mice you infect with Citrobacter, you don't see bacteria in feces, blood, or spleen after a few days later, day 10. But in NF-kappa B2 knockout, because they were not able to mount sufficient NF-kappa B response, they were not able to sustain inflammatory gene expression to an extent which was important. That led to huge amount of bacterial dissemination systematically. It actually spread all across the body. The mice died, right? They died. Wild-type mice was fantastic. They gave immune response. They survived. NF-kappa B2 knockout mice died. So what are we learning? That there are systems like NF-kappa B system where multiple signals are together integrated, and that leads to dynamical control, just accurate dynamics, which is required for efficient pathogen clearance. And finally, another thing, mathematical biology, system biology, this rocks. This provides for this additional understanding. One pathway, canonical pathway, you have negative feedback, you have premature activation stop, termination, that's not going to do work. But how do then in physiology accurately maintain this late activity? It appears you actually have multiple pathways, they crosstalk, and that allows for tuning of NF-kappa B duration. And another thing I'm always trying to implicitly say, that we always say, very strong response has happened. We think about the amplitude. Today I did not talk about it. Only thing I convey to you that duration matters. It's not how strongly you respond, it's how long you can maintain a response. You can be very angry and next day forgive that guy, or you can be a person who is not immediately very angry but maintain the anger for months or sometimes years. Both matters. All right. So here I will stop. This is the laboratory. We are talking about systems medicine, but I'm not going to really, overwhelm you. But see, the component of the laboratory is mathematical model, cell culture experiment, and animal. How putting them together, you will sometimes know about biology. Here I talked about immunology. Thank you very much. I'll stop here. All right.